student. Today I am going to explain the topic DNA. You all must have heard about DNA in your previous classes or in movies, but today I will discuss in detail about DNA. In DNA, D stands for deoxyribo, N stands for nucleic, A stands for acid, which is deoxyribonucleic acid, which is proposed by Friedrich Miescher in 1869. Now let's talk about DNA composition. It is a long molecule made up of a small unit of nucleotide. What is nucleotide? Nucleotide has sugar, phosphate, nitrogenous base. Here we can see this one is your sugar, phosphate, nitrogenous base. So nitrogenous base again divided into 4 groups, which is adenine, thymine, cytosine, guanine. What are this nitrogenous base? In DNA structure, you have a phosphate plus sugar bond, and here in between these strands we have ladder-like structure which is nitrogenous base. So we can see the adenine, thymine, cytosine, guanine in the ladder-like structure of DNA. Adenine, guanine always form a double ring structure which is called purines, and thymine and cytosine are the pyrimidine, which is single ring structure. Now let's talk about bonding between these adenine, thymine, cytosine and guanine. A always bond hydrogen bond. A always bond with hydrogen bond, which is double hydrogen bond. C always have a triple hydrogen bond with G. A always double hydrogen bond with T. C always triple hydrogen bond with G. Let's discuss about these bonding in this structure. If I have adenine, then I must have thymine here with double hydrogen bond. If I will have cytosine here, then I must have guanine in this side with triple hydrogen bond. And let's talk about function of DNA. DNA have a strong genetic information which transfer from parents to offspring or children, and it controls all activities of the cell, like in nerve cells and in muscle cell, blood cell. In all cells we have a DNA. Cells have DNA, cells have nucleus, and nucleus have DNA. Now let's talk about structure. DNA have double helical structure. This one is double helical structure, which is proposed by Watson and Crick. And it have a twisted ladder-like structure, which is nitrogenous base. And it has sugar phosphate backbone. This one is sugar phosphate backbone, which runs antiparallel. What is antiparallel? If your first strand will run from 3' to 5', then other strand must run from 5' to 3'. This mechanism is anti-parallel mechanism or directionality of DNA. And your nitrogenous base are forming a ladder-like structure, and these hydrogen bonds help to stabilize this DNA structure in the nucleus. And in each strand and in each twisted like structure, we have 10 base pair, 10 nitrogenous base pair. Okay, so we have seen that DNA is proposed by Friedrich Miescher, but double helical structure of DNA is proposed by Watson and Crick. And this is all about the DNA. And so thank you so much for listening me.